Oi, oi, you filthy swines! Get in here, sit down, behave yourselves. I'm going to read some stuff. Let's get on with it, I suppose. If you've ever watched science fiction, specifically space opera type science fiction, cryogenics is a big recurring theme, and some people, you may think they're misguided, have spent money to try and cheat death. Well, in this article on the Daily Mail, written by Cassidy Morrison, frozen brains reawaken in astonishing medical breakthrough. It seems more fantastical that you could freeze a person and then thaw them out and they'd be perfectly all right, but you never know. Maybe there's some kind of antifreeze. I know they pump these bodies full of antifreeze and all sorts of other chemicals before they freeze them. For decades, the concept of cryopreservation has been rooted in the realm of science fiction, a stable plot device for sending characters on voyages into the future. A seemingly insurmountable challenge has been freezing complex biological tissue, particularly the brain, without causing catastrophic, irreversible damage. The groundbreaking study has now taken a significant step towards overcoming this hurdle, successfully restoring functional activity in frozen brain tissue, an accomplishment that brings certain futuristic possibilities into sharper focus. The primary obstacle of cryopreserving brains has been the formation of ice crystals, which can cause all sorts of havoc. I hear the slower you freeze a brain or a body, the larger these crystals are, so you have to basically flash freeze them. As water freezes within the delicate cellular makeup, these crystals expand, puncturing membranes, disrupting the intricate network of neurons, and ultimately destroying the connection that underline thought, memory, and consciousness. Oh no! That old chestnut there. This destruction leaves thawed tissue incapable of any meaningful function. However, a team of neurologists at the University of Erlangen-Nuremberg in Germany circumvented this problem by turning to a technique known as vitrification. This process cools tissue so rapidly that it prevents ice from forming altogether. Really? And instead of crystallizing, the liquids inside and around cells transform into an amorphous glass-like state, preserving the tissue structure with all molecular motion effectively halted. You'd have to freeze it pretty bloody quick, I imagine, to do that. I know in the film Demolition Man, they stick the criminals into like a bath of water or some other liquid and they shoot some frozen particle or something into it, which instantly freezes them. It's science fiction, so don't read too much into that. The team applied this method to thin slices of mouse hippocampus, a region critical for learning and memory, cooling them to 196 degrees Celsius, about minus 321 degrees Fahrenheit, with liquid nitrogen. The samples were then stored in this glass-like state for periods ranging from 10 minutes to a full week. The true test came when rewarming. The researchers meticulously thawed the brain slices, then probed them to see if any indication of life remained. The microscopic analysis revealed that the delicate neuronal and synaptic membranes had survived the process intact. Further tests showed that the mitochondria, the tiny energy sources within the cells responsible for metabolism, were functioning without signs of damage, according to the latest study published in Proceedings of the National Academy of Sciences, abbreviated to no, abbreviated to PNAS. Oh, there we go. That's reality for you. The team was able to record electrical activity from the neurons. The cells responded to electrical stimuli in a way that was near normal, though some moderate deviations from unfrozen control samples. I had a pretty bad day or month or year or however long it took. Most importantly, the researchers observed evidence of long-term potentiation, LTP, a process involving the strengthening of synapses that is considered a cellular basis for learning and memory. This finding suggests that not only were the individual neurons alive, but some of the complex functional circuitry underlying cognition was also intact. To achieve this breakthrough, the German research team started with thin slices of mouse hippocampus, a region critical for memory. These were immersed in a potent cocktail of cryogenic agents introduced in steps to prevent shock. Once fully loaded, the slices were plunged into a copper cylinder cooled by liquid nitrogen. In this state, all molecular movement stopped completely for up to a week. Rewarming was just as critical as the freezing. To prevent ice from forming as the tissue transitioned back to a liquid state, the slices were warmed incredibly fast. at a rate of 80 degrees C per second in a warm solution, 176 degrees Fahrenheit. It's pretty warm. It's not boiling, though. You're not going to cook a steak with that, though. Once thawed, the potential chemical cocktail was carefully washed out in a way that prevented the cells from absorbing water too quickly and bursting. The team then attempted to turn an entire mouse brain into a glass-like state. Oh my goodness. Poor old mouse. A major hurdle was the blood-brain barrier, the brain's natural defense system. It lets water pass through easily, but blocks the large molecules of the sea pass. The researchers' solution was to alternate perfusing the brain's blood vessels with the protective chemicals and the carrier solution. This approach loaded the tissue evenly without causing catastrophic dehydration or later fatal swelling. After rewarming, the team put the tissue through a rigorous battery of tests to see if it had survived. They measured oxygen consumption to confirm the cell's powerhouses, the mitochondria, was still working. They used powerful electron microscopes to check if the delicate connections between neurons, called synapses, remained intact. And crucially, they inserted tiny electrodes to stimulate the cells and listen for a response. What's this? Stimulus number. Control. Okay, so that's a brain that's not been tampered with. Post-vitrification, post-CPA loading. So almost back to normal. In normal tissue, black and tissue exposed only to the protective chemicals but not frozen blue, response gets stronger with each pulse, a sign of healthy short-term plasticity. But in tissues that went through the full freeze-thaw process, red, this strengthening is muted. This confirms the change comes from vitrification itself, not just chemical exposure. Remarkably, not only could individual neurons fire in response to stimulation, but the complex circuits underlying learning and memory were still operational. The observations were limited to a few hours as the brain slices naturally degrade after thawing, and the work was done on thin tissue selections, not a whole living brain. But they said further up, you said up there, all the way up there, that you tried to do it on a whole mouse brain, but then you just kind of wandered on and forgot about that. So I'm assuming that poor old mouse brain was thrown in the trash. Poor old Squeaky. That was that. Oh, wonderful name here. Mrityunja Kothari, a mechanical engineer specializing in cryobiology, told Nature this kind of process is what gradually turns science fiction into science possibility. However, he added that applications such as the long-term banking of large organisms or mammals remains far beyond the capabilities of the study. Nevertheless, the implications for health and medicine are notable. This research opens new avenues for protecting the brain after severe injury or during disease, where inducing a protective suspended state could buy precious time for treatment. That's one of the themes in the movie AI, where the family that adopts the AI emotional robot has a son who is critically ill but held within some state of cryogenic suspension. And then he comes back and resents his robot brother and, spoiled the plot for you there. Sorry. It also suggests that researchers could potentially allow for the long-term storage of donor brains for research, or more realistically, other complex organs for transplantation. This study provides the most compelling evidence yet that its foundational science may be slowly building. It'd be a bit worrying if you donated your brain for study and then they just started prodding you and poking you and you weren't exactly dead. I know they're doing it to animals here, but at some point it's going to be done to people, imagine. Is it Walt Disney who has his brain frozen? And other famous people? I don't doubt that it's a possibility that you could eventually resurrect people in a certain fashion, but I don't believe it's realistic to imagine you could thaw these brains out. The best thing you could do, if your brain was frozen, you could go through some kind of destructive scan. If you could take slices of the brain and then record their structure and try and create some kind of simulation of their brain, that would be more realistic than trying to thaw the whole bloody thing out and then jump start it with some jumper cables and hope for the best. I think once you're dead, you're dead. And the best they could do is analyze the pattern that is you and have that run virtually within some kind of simulation. I think would probably be the only realistic solution, and that's crazy sci-fi. But I think we are heading towards a very crazy sci-fi world. I think we're already in a crazy sci-fi world. So I don't think we should put that kind of craziness on the shelf. I think some of these people who have been frozen may end up coming back in some capacity in the not too distant future, maybe. I mean, how many people have been cryogenically frozen? I bet there's quite a few. I bet there's at least a few thousand brains out there frozen. And if there's a way to scan through the contents of those and build some kind of emulation of that person, I think people will be down with that. Make sure you hit that like button wherever you are. But that is it for now. And the only thing I have left to say is Pigwig out.
Oink, oinkity, snort, snuffle, snuffle, squee, oink, oink, ree, squeal, snort, snort, oinkity, squee. Oink, oink, oinkity, squee, squee, ree, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 ree, 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 oh. Snort, snuffle, oink, squee, squeal, ree, snort, oinkity. Oinkity, oink, oink, snuffle, ree, squee, snort, oink, squeal. Oink, oink, oinkity, squee, squee, ree, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 ree, 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 oh. Oink, oinkity, snort, snuffle, snuffle, squee, oinkity, oink, oink, snuffle, ree, squee, snort, oink, squeal. Oink, oink, oinkity, squee, squee, ree, 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 snuffle, snort, squeal, squeal, oinkity, oink, snort, snort, squee, oink, 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 ree, 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 oh. Squeal, squeal. Snort, snuffle, oink, ree, squeal, ree, snort, oinky. Oinkity, oink, 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 snuffle, ree, squee, snort, oink, squeal. Oink, oink, oinkity, squee, squee, ree, 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 snuffle, snort, squeal, squeal, oinky, oink, snort, snort, squee, oink, 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 oink.